troubleshooting will help you to this thing, do your task. So we were doing what? We were doing, okay. So if you can remember that we were doing this again. We were doing what? We are doing a study to analyze the impact of three different types of mutations on a particular sequence, which is vitamin D3 receptor. Okay. So at first we tried to identify the receptor protein sequence from NCBI. Since NCBI is having some trouble, we are working on a different set of this thing, what do you call as database, which is basically maintained by Europe. Okay. So in Europe, this is a database which is giving you the sequence of vitamin D3 receptor, which is also for human beings. Okay. For human beings, it's the sequence of vitamin D3 receptor. Okay. Though it is not a typical FASTA, but it looks the same. Okay. So we'll use this sequence. We'll use this sequence, copy, and we'll go to this. Okay. Okay. So here what happens? So what is this sequence? This sequence is for vitamin D3 receptor. So let us calculate how many amino acids are there. Can you see? These are the single amino acid names. Okay. All there are 20 different amino acids. Each amino acid is identified by a letter. Okay. So can you tell me what is the first codon or the first amino acid which is present here? Who can tell me? Can anybody tell me what is the idea of the first amino acid? Which is, this is the N-terminal residue of the amino acid, and this is the C-terminal residue of the polypeptide chain. Okay. So there are how many amino acids? 10, 10, 10, 10, 10, 470, 474. So 474 amino acids are there. Okay. So if we consider the first amino acid as the first reading frame, then who can tell me the name of the first amino acid? Who can tell me? Can anybody tell me the name of the first amino acid?
Methyl.
This is methionine. Okay. It started with
Methionine.
M. So methionine is the initiation codon. So methionine is again, so we'll discuss a bit about methionine. So we'll discuss a bit about methionine. Okay. So we are having our sequence for vitamin D3 receptor. Okay. So what is the this thing of our vitamin D3 receptor? Let us consider this is our vitamin D3 receptor. Okay. So the vitamin D3 receptor is having an N-terminal domain and a C-terminal domain. Okay. The C-terminal domain. So N-terminal domain is the amino acid which is methionine. So this is consider that this is methionine. So this is methionine. So methionine is also known as, so we'll discuss a bit about methionine and on protein structure as well. So methionine is known as the initiation codon, or initiation codon means most of the proteins will have this amino acid as the amino acid number 1. Okay. And the code for this codon is AUG. AUG. That means here also there is a thing to discuss. Here also there is a thing to discuss. So AUG will be the N-terminal end, and here it is a C-terminal end. Okay. It started with the AUG. So in the mRNA or in the DNA, it is coded as AUG is coded as what? It is coded as TAC. So in the DNA it is coded as TAC. That means in the coding strand of the DNA, strand of double-stranded DNA. Whenever a TAC is there, it will convert into, or it will translate into AUG. Okay. And this is the initiation codon. And the C-terminal, there are how many amino acids? So there are 474 amino acids. Okay. In the what? In the vitamin D3 receptor protein in the vitamin D3 receptor protein. Okay, so now what we'll do is, okay, so this is the graphics. So now what we'll do is we'll copy the whole thing and add into the what you call tool. Okay, in that tool the default setting is this, but there should be a this structure. Okay, then only it will identify. So what happens, there are 474 amino acids. Now what we'll do, we'll try to figure out what would happen if there is a mutation on, the protein, and which is this, which is a missense mutation. Okay, so what we'll do is we'll design a mutation, let us say on the 6th position. So let us see what is the 6th amino acid. Methionine, phenylalanine, aspartic acid, valine, leucine, valine, leucine, leucine. Okay, so the 6th amino acid is 1, 2, 3, 4, 5, 6. 6th amino acid is valine. Okay, so 6th amino acid is valine. Okay, so valine, so I'll go and discuss valine a bit. So valine is an amino acid, V for valine. Valine is an amino acid which is, what is the character of the amino acid? The character of the amino acid is, it is non-polar amino acid. Okay, this is a non-polar amino acid. So this non-polar amino acid, we will mutate it. The 6th amino acid we have identified, which is valine. We will convert valine into aspartic acid. Okay, aspartic acid is D, and now we will see what is the result. Okay, so here you see, we told you that in missense mutation what happens, there is a single nucleotide polymorphism which leads to what you call an altered structure of the protein. Now this altered structure protein may be very distorted, or it may be a limited distortion. It may enhance the binding of the protein, it can also reduce the binding of the protein. So by this tool, which is a probabilistic classifier tool, we will try to figure out what is the impact of this mutation. What is the mutation? 6th amino acid has been converted in native protein. The 6th amino acid is a valine. Now we are doing a mutation. What we are doing, we are converting valine into an aspartic acid. Now we will see what is the result. Okay, now here you see grid gateway interface. Can you see grid gateway interface? It means that it has entered into the what? Grid computer of the Harvard University. Okay, now this is Harvard University Genetics Lab grid computer network. Grid computer is a big computer which is a kind of mainframe computer. Okay, now here you see the load is light, health is okay. So we'll refresh, we'll see the result is there or not. Or there is an error. We'll see what is the error. Okay, since the tool is free and for academic purpose, we'll see what is the error. Unable to locate protein entry. Okay. Okay. So one more time. No, it is not able to. Okay, we'll change it. Anyway, this is how this thing works. Since we have used a protein which is different from the usual one, means why? The database is different, therefore the format is also different. So it is not able to find out. Okay, one more time. The 6th amino acid, 1, 2, 3, 4, 5, 6. 6th amino acid is valine. We are converting it to aspartic acid. See, one more time we will give. Again the error is being given. No, error is again given. So the better will be to check for this usual database only. If it doesn't open, then what we'll do is we'll take the sequence that you have. You are having one protein sequence, right? That we downloaded lastly. Is there any protein sequence you are having? Can we download, or let us download it? Okay, fine. Okay, now we'll have our things done. Okay, okay. The sixth amino acid is a valine, and we'll convert it into an aspartic acid D. Submit query. Okay, so this is the result. See, so what exactly happened you see here. Okay, very nicely you see. So our protein was vitamin D3 receptor. Okay, so this vitamin D3 receptor has been identified as which is by means of a different this thing, what do you call as a database, which is an European database, not as user friendly. So what we did is we identified the sequence, and we are running this particular software, where we use the grid computer network of Harvard University, and we found that the impact of the mutation. And what was the mutation? We mutated the sixth amino acid from valine into aspartic acid, and this is the result. So how the result has been given, you see. The result has been given as possibly damaging with a score of 0.923, where the sensitivity is very high, 0.81, and the specificity is again very high, 0.94. So what does it mean? The sensitivity specificity is very high, the score is also very high, this thing here, and therefore it indicates that the impact of the mutation, that is which mutation? Sixth amino acid converted into the sixth amino acid in the normal protein is a valine. We have converted it into an aspartic acid. We have done it randomly. Then you can ask me, sir, on what basis you have identified the sixth amino acid? We can do anything, because I know that the sixth amino acid mutation of this particular protein is dangerous for the receptor. Therefore, I have given you this this thing. You can either use any of the what do you call you can play with the whole thing, and you can eventually do any sort of mutation. Okay, but there should be some biological background information regarding this. Okay,
Sir,
so what happens? Just hold on. So what happens whenever there is a mutation of this sort? It will lead you to a massive distortion in the protein. And what will be the impact? The impact will be a massive destruction of the structure of the VDR receptor. Okay, and by and large, since the result is what possibly damaging. So since it is damaging, what does it mean? It means that it will not able to bind vitamin D3 at all, and therefore there will be complete imbalance of what calcium metabolism, because vitamin D3 is very important for calcium metabolism. We already know the role of calcium in human beings. Okay, so these are the things you can remember, that there is a calcium channel on the surface. This calcium also helps in muscle contraction via actin myosin filament. This in turn is dependent on a cellular structure which is called sarcoplasmic reticulum. So calcium is also the number 4 factor of blood coagulation pathway. So here I should mention, in blood coagulation pathway there are two types. One is intrinsic and extrinsic. Intrinsic and extrinsic pathway. Extrinsic pathway. Okay, and there are 1 to 13 factors needed for completion of hemostasis. Hemostasis means blood coagulation. Okay, so these ideas should be there. Okay, just hold on. Yeah. Okay, so these are the idea. Okay, so these are the things you should remember. Okay, you can do the hands on by yourself.
So I have a question.
Yeah, tell me.
So what is the sensitivity and specificity high? What does this mean?
It means that it has used a huge database, and in all the database it has been seen that whenever there is a mutation of valine to aspartic acid, it leads to some kind of distortion in the protein. Okay, so specificity means exact, it identified the mutations not around the sixth amino acid, but on exactly on the sixth amino acid. Many a times what happened in homology modeling class, you might have understood, identified that the sensitivity and specificity was very low. So what does it mean? It means that the coverage of the protein is also very low. For example, we are having 400 amino acids, but in the structure we are only having 60 amino acids. Okay, so 340 has been lost because I could not find any suitable, what you call, template. So in that case, the sensitivity as well as specificity both reduces. But in this case, what happened? The database, the algorithm has identified huge amount of data which supports the fact that if at all there is a sixth amino acid mutation on this protein, that also from aspartic acid to valine, it will lead to some kind of overall destruction of the metabolic pathway itself.
What is the score? 0.923 means.
This is an inbuilt score which depends on the sensitivity and specificity. So as you can see, sensitivity and specificity both are more. So if any of these two are different, then the score might have been different as well. So you can play along, you can yourself find out these answers. You can use the tool that I have already given you, and by yourself you will find it out that the score will be reduced whenever sensitivity and specificity also reduces. Okay, so any
Okay.
more doubts? Okay, since the previous usual this thing has not worked, we had to use this tool. Okay, so these are the info. Today I'll share one more research study which will help you to think about various these things of what do you call as antibiotic resistance. Okay, in bacteria. Okay.